Good afternoon. Thank you very much for joining us today here in person and on your screen at home or at work. It's a great honor to welcome such a distinguished panel. We have 5 panelists here in person and remotely, and I will introduce each of them before their talks. My name is Claudia Langenberg. I'm Professor of Computational Medicine at the Berlin Institute of Health here at Charité, and MRC investigator and group leader at the University of Cambridge in the UK. My role today here is to moderate our expert panel. The session is 90 minutes long, and we will have one brief statement session by each of the panel members, followed by a joint discussion. I very much look forward to the presentations and a hopefully lively debate, including questions from people either here in the audience or remotely. Those questions can also be submitted if you join remotely via Zoom. Allow me to just start and kick off with a very brief notes or some context. It's been 20 years since the completion of the first human genome sequence. And this, together with the development of new genotyping and sequencing technologies, has led to an amazing acceleration and explosion in the number of discoveries related to the genetic basis of rare and common complex diseases. However, there's a perceived disconnect between the rapid success of genomic science and the expected or promised paradigm shift towards genomic or personalized medicine. There are already very clear examples of clinical applications for rare pathogenic variants, specifically Mendelian disorders and cancers, infectious disease control and drug development. However, applications for the clinical management of common complex diseases or targeted approaches or genetically targeted approaches to screening and or prevention are few, and they're not systematically implemented. We have gathered genomic data on millions of individuals and patient study participants. And again, much of our knowledge stems from selected populations with clear limitations with regard to the transferability of insights derived from predominantly European descent populations and a lack of focus on diseases of greatest global and global South relevance. Sharing of data and results distinguishes the genomic community that I'm proud to be part of. And long before the current pandemic started, it was self-evident that rapid data sharing provides massive benefits to science, scientists, and society. And again, free and open access is not yet universally implemented, involves a patchwork of repositories, can be delayed, involve tedious, onerous processes. And now that we have the ability to generate global genomic data of unprecedented detail and size for collaborative science, and this is very exciting, but it's clear it brings new challenges in terms of data standards, governance, and computation. So we have come here together today to reflect on the successes and initiatives that our panel members have initiated and hear about actions that are needed to address some of the pressing challenges, some of which I just outlined, and that we will hear about, and stand in the way of delivering the promise of genomic medicine in a global context. And before we start, I would just like to very quickly confirm that Bartek Knoppes and Charles Rotimi can see us and hear us.
Yes, we can.
Great, welcome. So good to see you, thank you. So without further ado, I'd like to introduce our first speaker. This is Professor Ewan Birney, who's Deputy Director of EMBL, the European Molecular Biology Laboratory, and Director of EMBL's European Bioinformatics Institute in Hingston, Cambridgeshire. He's Non-Executive Director of Genomics England, Chair of the Global Alliance for Genomics and Health, and was the analysis lead for ENCODE, set up to identify and analyze the complete set of functional elements in the human genome. And in addition to his many awards and distinctions for his contributions to genome analysis, interpretation, and computational biology, I would like today specifically highlight him as a driving and vocal force for open science and data and information sharing, way beyond human genomic data in the Ensemble Genome Browser, which he co-founded. Thank you so much for joining us today here in Berlin. I really look forward to your thoughts.
Great. Perfect. Well, thank you very much, Claudia, for that great introduction. It's lovely to be here in person in Berlin. I really love coming to Berlin. And just to also acknowledge that I think 10 years ago or 20 years ago, somebody from my standing perhaps wouldn't have been invited to this conference. And that was because we considered basic research to be a long way away from clinical applications and clinical practice. And that's not what is going on at the moment, that we are seeing now basic research, fundamental research, and clinical practice all converging very often around data. So let me start by just introducing the organization that I'm Deputy Director General of, the European Molecular Biology Laboratory. It is a sister organization of CERN. We're headquartered in Heidelberg, Germany, and we have 6 sites across Europe. And in this case, the UK, thankfully, has not left this European treaty. And so one of our sites, EBI, is present in the UK. So I'm British, I'm European, very proud of both. Further, the reason for that site just south of Cambridge, EBI, in the 1990s was set up to enable data sharing. Molecular biology has had a long-standing tradition of sharing data, arguably from the very start when Drosophila genetics was being created in the 1900s, but certainly by the 1960s, where Margaret Dayhoff's unique work in Washington, D.C. started to collate protein sequences for everyone to use. This was followed by research databases, the PDB, and then ENA, the European Nucleotide Archive, GenBank, and DDBJ in Japan over the 80s. And that collaboration, INSDC, has been the foundation of data sharing in the biomolecular space before the internet and continues to this day. Now, it's important, I think, here to separate out the different types of data that we share. We share molecular data about non-human data sets. We share molecular data, but with strict controls on human data sets, and I'll talk about that in a moment. And we also have to think carefully about the metadata that is associated with both of these different types of samples, something which I'm sure Sharon will talk about in the future. Viruses, bacteria, mice, wheat don't have ethics. And so there's a very different class of problem when we think about data sharing for those organisms compared to humans. And in those cases, the very open aggregation of data sets that molecular biology has relied on from the very start of its foundation, leading to the Human Genome Project, has been a great model. And that includes for things like antimicrobial resistance, for plant pathogens, for model organisms like mouse and Drosophila, and actually also for the reference human genome, where individuals generously gifted data only for the reference data, and the only information we have for those individuals is that they are human and they were alive once. So we carry no other information around for those reference human data sets. And in the COVID-19 era, we worked together with colleagues from the European Union, from the European Open Science Cloud, and from Elixir Europe to develop the COVID-19 data portal. This is a portal which aggregates data on the COVID-19, on SARS-CoV-2 across the world. We encourage co-submission to the public health GISAID database alongside submission to this fully open INSDC database in the COVID-19 data portal. And we have developed specific ways that public health individuals and countries can control when and what metadata they publish alongside the viral sequences. And so a country in Nigeria or a outfit in, say, Catalonia can decide when they publish their data openly to the world and with what associated data sets, metadata. And those are called data hubs. I'm very proud to say that with our partners, we have the simply largest amount of fully open access data on the COVID-19 data portal. And that is not just the viral sequences, but also the RNA that is produced from the virus, the protein structures that come about it, and chemical molecules which may interact with the different components of the virus. So an integrated data set to look at. And I encourage you to go and look at this information. Now, one thing that is often thought about here and sometimes keeps research at some distance from clinical work is to consider research only about fundamental discoveries and not necessarily about things that can really impact people's lives. And I just want to emphasize how I think misguided that can be, that one needs to bring these data sets together to allow researchers to have insight. And often those insights are truly serendipitous. So this is a very old example, but still remarkable. When researchers first sequenced the Plasmodium genome, the parasite that causes malaria, they were extremely surprised to find matches to plant chloroplasts. It was not thought at the time that the complex parasite Plasmodium was actually a sort of degenerate plant, apocomplexan. It was hinted at that they had complex organelle structures. It was only when the genome was sequenced did they realize this relationship between Plasmodium and plants. And in fact, that story repeats in the future when people investigated potato blight, not a medical concern, but a very big agricultural concern. This was considered to be a fungus, a non-traditional fungus. For many people, it looks like a fungus. It propagates like a fungus. When you look at it, it has all the phenotypic characteristics of a plant fungus, but in fact, it's an oomycete, similar again to the parasites from the Plasmodium. It is a degenerate plant. And so the targets that you choose to combat either Plasmodium or oomycetes, in this case for potato blight, are very different. And that is an example of the sort of serendipity that comes from having aggregated data for research purposes. Now, I just want to flip to the other side of this work, which is responsible data sharing of human genomes. It has been remarkable how much cheaper, quicker, and more portable DNA sequencing has become around the world. And this is a boon to infectious disease genomics. I'm sure Sharon will mention that. And also to human genetics. And Claudia mentioned that I'm a non-executive director of Genomics England, which is the NHS's way of delivering in the UK genomic medicine. I've also advised Genomic Denmark, Finland, and Plan France Genomic in France in my role as advisor to European endeavors. And in all of these cases, we are seeing the benefits of sequencing humans for clinical practice. The strongest areas of that are rare diseases. Although it's called rare disease, about 2% of children are born with a rare disease, very often can be identified at birth, and around 30% of those children can get a confident diagnosis via genomic sequencing of them and their parents. Of those 30%, only a handful have transformative changes in their diagnoses, but nearly all of them have an improved prognosis due to accurate diagnosis. And furthermore, there's a huge amount of worry relieved from the parents and from the healthcare practitioners by getting a confident diagnosis of these genetic diseases. And we can see that growing in the future. But what about access to this data? Should we have access? Well, those concepts of serendipity and research are as valid in human, but of course humans are ethically protected. They come in societies, we have rules and legal processes, and we cannot aggregate this data globally in the same way that we can aggregate data from non-human species. And I just want to fast forward now to the future, and this is something that I know I share with Bartha Knoppers coming up, and why we founded the Global Alliance for Genomics and Health. And this is to recognize that in the future, now and in the future, data on humans, large-scale data on many human patients and citizens will be kept nationally under national rules, national legislation, with individuals making access decisions that come from those countries. What we should hope to happen or expect to happen is that researchers who are doing responsible beneficial research for everyone can get access to those data sets by applying in simple and easy ways. And just as Bartha and Charles are visiting over Zoom here to this conference, researchers can visit Denmark or Nigeria or Malaysia remotely in a responsible way. Now, that future is what the Global Alliance for Genomics and Health, GA for GH, please check out our website, is aiming for. And I'm chair of that organization. It has a global community making standards for this. And that's standards not to control access. Access will always be controlled by individuals in the different countries. It is standards just like the web standards that this presentation is being beamed over at the moment to allow technical access, given that you have had authorization. A big part of that is harmonizing the way we think about ethics and access and legalization, and I'm very lucky that I don't have to fit that into this talk because Bartha is coming up. So I just want to return to my original theme, say thank you to Claudia, to the other members of WHO here for inviting me, because I feel it's very, very important to bring colleagues like myself. We come from a research background. I was one of the kids who first annotated the human genome and put it together back in the 2000s. I have gray hair now, but I'm still as enthusiastic about making these technologies radically beneficial to everyone on the planet in a responsible and sharing way.
Thank you so much, Ian. If I may have one follow-up question. As you started out when you spoke, it's a really great term phrase, convergence of clinical and basic science around data. And I just wonder if we think about that this pandemic has acted as a catalyst to kind of bring this together, what is the one lesson or opportunity that you think we need to actually make sure we secure to continue for other areas of science?
Well, I think that's an interesting, I think we all have discovered that we have learned more through this pandemic here. I've definitely learned two things. The first thing is to respect my clinical colleagues and clinical practitioners. Healthcare and healthcare delivery is immensely complicated, and it is a enterprise and an art. And the second thing is that we, when we want to make progress, very often we've got to leave our egos at the door, so I have to learn to do that sometimes.
So as Ewan already announced, it's my great pleasure to now welcome Professor Bartha Noppes, joining us remotely. Welcome. Professor Noppes is Professor of Law and Director of the Center of Genomics and Policy at McGill University in Canada. She's a world authority on the ethical aspects of genetics, genomics, and biotechnology, and with major influence on international policy making, for example, by drafting UNESCO's Universal Declaration on the Human Genome and Human Rights, leadership of the ethics work of HapMap and the 1,000 Genomes Project, the presidency of the International Ethics Committee of the Human Genome Organization, or HUGO, just to name a few. Thank you so much for joining us today and contributing your experience and views here on the ethical and governance issues that lie ahead. Welcome.
Thank you, Claudia. Hello, everyone from Canada. It's a great pleasure and privilege to be here with you. I've divided my very short presentation into three parts. Challenges, I call them challenges, not barriers, opportunities, and then I too will opine on the influence of the pandemic on data sharing. It's only been 40 years, as Ewan has pointed out, that the great genomic adventure began. As Claudia, however, pointed out, even today, 40 years later, data sharing can be both tedious and onerous in terms of the processes that have been put in place. And to understand why some of the ethical legal norms that govern data sharing or data accessibility or data protectionism, as I call it, have to look at how policymakers and lawyers, what their context is in which they make laws and policies and regulations. And we can only point to a few examples, but even though biobanks that started in the early 2000s, so on, started seeing data science as an infrastructure or data sharing building data resources at a large national level, mainly in order to have statistical power and significance necessary for stratification. And so for epidemiological studies that would allow us then to do resource allocation in healthcare systems. Still today, less within countries, but certainly within Europe and also internationally, we still have many challenges. Some of them stem obviously from the scandals, like the Cambridge Analytica scandal, thefts, leaks of data. The notion that genetic information is somehow different from other medical information, and so is sensitive and must have a higher degree of protection rather than normalization and integration into care. Conservatism, protectionism surrounding re-identifiability. Somewhere, somehow, someday, someone, some young 14-year-old hacker would be able, by putting together different data sets that are publicly available, to re-identify an individual. This has led a lot of policies to tend towards favoring anonymization, which of course is inimical to being able to use the genomic research in the clinical domain because of the inability to link and to go back to persons if data is encoded and encrypted or protected in other ways than anonymization. Also, the failure to include subpopulations and to have a diversity of populations in our databases has really attracted a lot of attention. And there are now projects beyond the HapMap, beyond the 1,000 genomes, that are attempting, such as the Pan-Genome Reference Consortium, to be more representative of communities, countries, peoples, and hence genomes around the world. But the big, I will use barrier here to international data sharing is obviously the adoption of the General Data Protection Regulation, which came into force in May 2018. Now, this is a European regulation, but as a regulation, it has much more effect, legally speaking, than a directive. And certainly within Europe, that is being felt. But it had an unintended extraterritorial effect, as we say in law as well. It actually prevented and stopped, halted, and redirected data sharing between Europe and North America, South America, African continent, and so on. Because if you couldn't receive data because you weren't considered to be adequate with the general data protection level, now legally required in Europe, you would no longer be able to share. So we saw the attempt to start pooling data in large consortia going back to federated and distributed models. We can talk about that during the question period. So opportunities, let's get on to a better topic than all the difficulties and the sociocultural context in which laws are made. As Ewan mentioned, in 2013, the Global Alliance for Genomics and Health, of which I'm one of the founders, adopted a framework, the framework for responsible sharing of genomic and health-related data. So that bridge between the genomic and the health-related was laid down in a vision, I could say, of the Global Alliance GA4GH. And contrary to most international treaties, declarations, and so on, the framework for the Global Alliance was founded on a human right that already existed in 1948 in the Universal Declaration of Human Rights, and then was repeated in a treaty that made it legally binding on 196 countries who signed and ratified. And that human right was the right of everyone, and that's everyone, to benefit from science and its applications. And it's what we call a Cinderella right. It had been lying there since 1948. And we wanted to use this human right in the framework of the Global Alliance to catalyze it, to catalyze the freedom of science, but also of citizens around the world to benefit from science and its applications. making it an actionable legal right. And so the Global Alliance built not only the framework, but policies, procedures, and tools, some of which I understand, others which I don't, but also resources, Matchmaker, the BRCA exchange, API beacons that go looking for alleles and so on. So it goes all the way from the framing on principles to the policies on security, privacy, ethics, and so on, to the actual tools to activate this human right. And our final actually we're working on it right now, so it's interesting that Ewan mentioned it. We're now actually working on clinical genomic consent. So not your 25-page research consent. We're looking to build a sort of a harmonizable template that can be used in the clinic to make sure that genetic information becomes part of the medical record. Now, maybe one day we won't need such a separate very short consent, hopefully. But right now, since genetic information is still considered sensitive, the projects such as in the UK and Australia and in the Nordic states are adopting this approach to genomics in the clinic. So finally, with the arrival of the pandemic and all the challenges that it has placed before us, Ewan mentioned the COVID-19 data portal, COVAX for vaccine distribution, GSED, the global initiative on sharing all influenza data. So it's brought the global community together out of necessity, but I think we can really build on the lessons that it is teaching us and the new kind of ethical legal structures that we can put in place, hopefully, not only for the pandemic, but to change data sharing from now on. And the first sort of opportunity that the pandemic brings is that it sort of resurrects, revives, or it's a renaissance of the public health, public interest rationale underlying research. So move away from continually using individual, explicit, specific consent for any use of genomic and health data, more in seeing this scientific endeavor as part of the under the public health rationale, but in the public interest, and seeing data sharing and global data sharing of data as global public goods. And so we move away from the individualist privacy, me, myself, and I ownership model to a model that doesn't say give everything away, but that there should be a minimum amount of exchange of necessary genomic and other medical information in order to understand the pandemic, to prevent future pandemics, and to move forward for the future. And I have two ideas on how we can do that. There is a movement towards this already. In 2017, the OECD adopted an international health data governance framework. In November, so next month, UNESCO will be adopting its recommendation on open science. So you can see that the large international bodies are getting on the data sharing bandwagon. So I'd like to conclude with two possible futures for global data sharing. One, and I hope I don't have to be a part of it, is to draft a pandemic treaty. I really think in the long run, we need a very secure, legally structured, ratified by countries, which makes them accountable. They have to report in what they've done or not done with what they've signed and ratified. And I think a pandemic treaty in the long run is necessary to ensure the future of global data sharing. But in the meantime, what I would do like tomorrow is to reinforce the international health regulations of the WHO. I would add new regulations, I would provide more funds, and I would put in stricter sanctions because it's already there. So it's the structure, the tools, the obligations, and so on only need to be funded and reinforced. So I'd like to conclude by saying that the idea then is to move away from the model of individual privacy risks and individual specific consents in every situation. I'm saying it is necessary for research and so on, but it's not necessary for public health research. The powers are already there. So if we move away from individual privacy risk language to collective health global opportunities, I think would make it possible to cross or to build quality health research on a global scale. Thanks.
Thank you so much. What a fantastic build on some of the themes that you started. And I'd love to discuss what you mentioned, the federated approaches in the discussion. And if I just may too, you started out with the context of the law and regulations. And then later on, you mentioned the clinical consent or minimal clinical consent. And I just wonder whether you could comment on how is this context dependent, or can we move to a kind of more universally applicable clinical consent for what you named the kind of minimal information people should sign up to share?
Yes, because of the continuing sensitivity of genetic information, we haven't sufficiently normalized it so that people look at it like blood pressure or other health conditions, cancer, or the health authorities, the states, the ministers of health in different countries are still saying, if we start doing genomic testing in the clinic, where it's necessary for diagnostic purposes, I'm not talking screening programs here, that's a different public health approach. If we're going to use it in the clinic, we still need to have something signed. We still have to tell people that it's familial, as if they don't know. We still have to tell people that we might find things we aren't looking for, which happens in many cases of ordinary medical tests. We still have to tell people that there might be commercialization one day. I mean, really? Don't people know? So I don't understand it, but the health authorities would like to have that kind of sort of notification, I call it, of patients, that this is the way it's going to be when you have a genetic test. If the doctor thinks, or the health professional thinks it's recommended in your case in order to have a better diagnosis. So it's a step. I probably should use notification more than consent, but there are countries that still want a signature in order to sort of calm down in this integration process.
Great, thank you very much. I'd like to now introduce our next speaker remotely. We are joined by Dr. Charles Rotimi, joining us from the US. He's just been appointed as scientific director of the National Human Genome Research Center, part of NIH, or the US National Institutes of Health. And before that was director of the Trends NIH Center for Research in Genomics and Global Health. He founded the African Society of Human Genetics and has been instrumental for pushing population diversity and ensuring that African populations are better represented in genome research, leading the African component of the International HapMap project and launching the Human Hereditary and Health in Africa, or H3 Africa initiative, a 70 million collaboration between the NIH and the Wellcome Trust. First of all, congratulations to your new role. We are really honored to hear your strategy for genomics to benefit human health.
Yeah, thank you very much. And I'm excited to be a part of this discussion because of how critically important it is. I just want to make some, high-level remarks, and I will try to make about 4 points, in my talk. The first, sort of high-level comment is that achieving the promise of genomic medicine, a more comprehensive understanding of human history, how we are related, and how we migrated to different parts of the world, will really depend on the fact that we need to engage different ancestral populations, and we need to do work in different geographical locations around the world. Just as important is that achieving the promise of genome medicine or genomic medicine will depend on the equitable sharing of global genomic data and technologies. In this regard, I view data sharing, global data sharing, as the norm. That is where we should start from. That is, we should all agree, excuse me, that there's huge benefit in sharing data. I think once we make that agreement, then we now say, what is preventing us from achieving that, norm that all of us believe that is really where we are going to be if we truly want to reap the full benefit of biomedical research and genomic medicine, specifically. So, but we have to do this data sharing in the context of full respect from where the data is coming from, in terms of the population, the scientists, and also in terms of the resources and benefit that is going to come out of it. So I also want to say that not sharing data should actually be regarded as unethical, because we are wasting resources. We are wasting the contributions of human beings to biomedical research, and we are indeed hindering our ability to actually address world problems, together. So I think we have to look at it from different point of view. So that is really sort of the first high-level comment I want to make. But for me, from my experience, as you heard during my introduction, that I have actually been a very strong advocate for the lack of representation of diverse ancestral background in genomic research. This problem is well known, and the scientific and ethical limitations are also well appreciated, or increasingly getting appreciated, including misdiagnosis due to inadequate reference databases. If you are trying to understand the pathogenicity of a variant, then you need to be able to represent the global distribution of human genetic variations. Otherwise, you may indeed be arriving at the wrong public health decisions in terms of the distribution of that variants, or whether the variant is actually pathogenic or not. And also, having access to global data will also enable you to understand the genetic background in which these particular variants may be acting. So those are, again, very, very important reasons why I think we have to really study and share data across global populations. We also know that there is serious implication for drug metabolizing enzymes. If we do not fully appreciate the distribution of drug metabolizing enzyme, we may actually be compromising the health of different people. And the example I want to give is abacavir. There's a HLA variant in terms of hypersensitivity to this HIV drug. When you look at the distribution within African populations, the Maasai in Eastern Africa have one of the highest rates of these variants in the world. Whereas when you look among the Yorubas, the frequency of this variant is almost zero. And then in Asia, the distribution is also quite variable. So if you were looking at African as one unit group, you may indeed be making the wrong public health distribution because of these huge differences in allele frequency. So again, the major point here is that we need to engage all human populations if we are truly going to be able to make good public health decisions and also therapeutics, decisions. So that is why I say we, have to establish the H3 Africa program and the African Society, as you heard during my introduction. But the limited data from African ancestry is especially troubling because Africa is the birthplace of modern humans, and it harbors the greatest diversity. If science was actually rationally done, and you want to actually have, in the context of limited resources, in terms of sequencing human genome, you will start from Africa because that is where you are going to have the highest gain for your money. But the funding of science is not really that rational. Is governed by where the money is coming from and all of that. So I think we need to look at global context. And I think The previous two speakers alluded to COVID as another example of the lack of appreciation of global populations in terms of making success in how we develop vaccines and how we administer vaccines and who is benefiting from it. Now, there is a substantial proportion because of this history of migration out of Africa. And so sometimes the point I make is that we are all Africans beneath our skin. And that's because if you trace your ancestry far enough, you're probably going to end up somewhere in Africa. Because all of us, modern humans, migrated out of Africa about 100,000 years ago to populate the rest of the world. But what is important and why I think we need to focus and do a systematic sampling of African populations is the fact that the subset of humans that migrated out of Africa only took a subset of the variation that existed at that point. So there's a huge amount of variation that only you can study in the context of African populations. So if we do not study that very well, we are going to indeed compromise our ability to truly understand biology and genome medicine. We've seen an example of this in terms of something like the PCSK9 in treatment for the so-called bad cholesterol. Although it was because of the higher frequency in African ancestral populations, the discovery was possible. But we now use our understanding of this particular gene in the context of treating cholesterol in other human populations. So the fact that you are understanding it in Africa doesn't mean other parts of the world are not going to benefit from it. Okay, that's another point I really would like for us to take away in this conversation. Also importantly is the lack of infrastructure. I think it's a hindrance to our ability to share data. The technology that we are currently using is expensive. And a lot of low to medium income countries are not able to afford this technology. So there needs to be a global strategy to develop technologies that will be friendly, that can accelerate our ability to share data and also to manipulate the data. I participated in HapMap with Bata and others. One of my fears in doing that work was the fact that, yes, it's going to be publicly available, but because of lack of data across African continent, that the scientists in that part of the world will not be able to use data despite the fact that it's publicly available. So publicly available does not mean that there's equitable access to that data and utility. So we need to address that, that bottleneck. Also, poor healthcare infrastructure does represent a real roadblock to our ability to gain, from genome discoveries. How are we going to be able to do precision medicine in an environment where the healthcare infrastructure is not even functional? So we need to look at this from a global perspective, because those populations are going to feel like they are not going to benefit, therefore they don't have any reason to share data. So those are some of the things I really want to put on the table. And COVID, I think, poured gasoline on the problem of lack of equitable access around the world, and even within nations. And we have to use this example to do better. Thank you very much.
Thank you very much. And you highlighted an interesting disconnect between rational science and rational funding. And if you consider that certainly some of the sequencing efforts in the UK are industry funded, yet the data will still be accessible to all, or in theory, at least as a first step, to all. What do you think is really needed to address this, or at least go some way of addressing this?
I think we have to show what I'll maybe put under the umbrella of global citizenship. We have to understand that, especially in the context of infectious diseases, but even outside of that, we have to fully understand that we would not appreciate the scope of genetic diversity of if we do not study our human population. And therefore, even if we are trying to solve the problem in Europe, we may not fully be able to understand that without studying populations in other parts of the world. So for example, we know that there are populations in South America that have, over the years, because of high exposure to arsenic, they have mutations that actually help them to metabolize arsenic. That is fundamental to our understanding of biology. You probably cannot get that from doing work in the US or in the UK. So I think that's really what we need to look at ourselves as global citizens, and that we have a common objective, and that is to improve human health. And we have to go beyond national boundaries, to be able to do this. And we have to be true to ourselves that even within the UK, I look at the distribution of the UK Biobank, for example. It's a wonderful global resource. But if you look at it in terms of ethnic representation, you still see limitations, there. And I think that is really what we have to focus on to make sure that we truly are looking after each other. And if we do not do that, you will see continuous resistance to true global data sharing.
And as you said, indeed, diagnostic errors and misjudgment of drug metabolism and so on across, many, many very large populations. So thank you very much for the answer. Thank you. I'd like to introduce our next speaker, which is Professor Sharon Peacock. She's Professor of Public Health and Microbiology at the University of Cambridge and an expert on microbial sequencing, including pathogens such as MRSA or methicillin-resistant Staph aureus, which is an important cause of difficult to treat healthcare and community-acquired infections. She was Public Health England's Director of the National Infection Service and has established and is Director and Chair of COG UK, the COVID-19 Genomics UK Consortium, a pioneering collaboration set up in March 2020 for rapid whole genome sequencing of SARS-CoV-2. COG UK has been absolutely instrumental for understanding viral transmission and evolution and informing public health responses in vaccine development. As of today, and I just checked this morning, it has sequenced 1,213,491 viruses. Thank you, Sharon, on behalf of all of us. Please share your thoughts on this globally uber relevant area of genomic science and medicine.
Well, good afternoon, and thank you so much to Claudia and the other organisers for inviting me to speak today. It's a real pleasure to be here. So early during the COVID-19 pandemic, people around the world recognised the potential value for rapid and large-scale genome sequencing of the SARS-CoV-2 virus to inform public health and also inform drug and vaccine development. These are actionable information. So really from the early days of the pandemic, it was really a question of when rather than if SARS-CoV-2 variants would emerge that had changes in their biological characteristics, which in turn changed the way the virus interacted with us. So these include, as you well know, increased transmissibility, increased severity or course of clinical infection, and the potential to evade natural or vaccine-induced immunity. So in the UK, in the early months of the pandemic, we sought to establish a system that generated sequence data for public health agencies to use in identifying and monitoring variants as they emerged and spread. We were not alone in this. Many other countries have developed such capabilities. Now, in the UK, we aim to be inclusive, and as a result, the consortium was able to bring together numerous universities, the 4 public health agencies of the United Kingdom and the Wellcome Sanger Institute, which became networked with NHS testing labs and high-throughput national testing labs. So in this way, we established a hub and spoke model for distributed sequencing with centrally organised logistics and operations for moving samples to the appropriate hub. We were fortunate in terms of data storage and analysis to have access to the cloud infrastructure for microbial bioinformatics, or CLIME, which housed a data management system and was further developed to contain the necessary analysis tools to support our efforts. So collectively, you've heard that in the UK, we've sequenced well over 1 million SARS-CoV-2 genomes. And I'd like to really thank and pay tribute to the 600 consortium members who've actually generated that data. They've done that largely as volunteers, and it's been the most extraordinary team effort. Now, underpinning our activities is a shared values of transparency and open access data release. And so we've shared our genome data with others as soon as possible through the global database GISAID and through the ENA and elsewhere. Now, thinking about the wider context of sequencing beyond COVID, prior to the pandemic, pathogen genome sequencing was already used by wealthy nations for a variety of use cases to really generate actionable information for public health. So, for example, sequencing pathogens associated with foodborne associated outbreaks such as salmonella or listeria has really become routine in many countries to help detect and investigate outbreaks. Sequencing is also used for TB. This can predict the presence of multi-drug resistance and can lead to more rapid administration of effective treatment for the patient. Sequencing has become established for HIV, particularly early in the course of infection, to detect the presence of changes in the virus that indicate resistance to antiretroviral drugs. It's also used during the treatment of other viral infections, such as hepatitis C. So coming back to the pandemics and looking ahead to the future, sequencing will be used to examine the reservoirs of novel viruses in the animal kingdom, including in bats, so that we can better understand our future risk. We will use sequencing again to detect and better understand the next novel virus that emerges and crosses over into the human population. So sequencing is likely yet again to prove crucial in vaccine development and monitoring for pathogen evolution and evasion from vaccines and drugs. So in short, sequencing has become a vital tool to detect, treat, and contain outbreaks both now and in the future. We've really broken through a glass ceiling that existed certainly in wealthy nations, where the value of pathogen sequencing has been fully realized and embraced by policymakers, politicians, and the public. But something that the COVID-19 pandemic has really demonstrated so very clearly is the deep and troubling inequity in access to modalities that can protect the health of nations. Now, the prime example is vaccines, but the same is true for sequencing, so that a country may have no or limited knowledge about the variants that are circulating. This is troubling for several reasons. First is the ethical standpoint. It's only fair that everybody has equal access to an understanding of what lineages are causing infection in their country. This will become increasingly important if variants of concern emerge that fully dodge our immune system. Second is a more strategic control point. Now, if new variants of concern are going to emerge, they're likely to do so in places with high rates of infection and transmission. With our interconnected world, we have seen repeatedly that it takes a very short time for variants of concern to travel around the globe many times over and infect people across the planet. Now, for a new variant of concern to emerge, it will need to be fitter than the Delta variant, which is already highly transmissible and partially evades our immune response. So it's reasonable to postulate that a new variant of concern that can outrun Delta would be even more transmissible and or have greater immune evasion properties. However, with low sequencing generation for positive cases being the norm in many countries, its early emergence and spread may go entirely unnoticed for a period of time. Only through the international generation of large-scale sequencing data will we be able to comprehensively monitor for variants whose increasing prevalence suggests cause for concern. Furthermore, we need to strengthen the science pathways that connect genomics with modelling and lab experiments to determine whether a variant under investigation with a question mark over it should be classified as a variant of concern and prioritised for public health responses. This means strengthening not just sequencing, but also virology and immunology worldwide. We're not where we need to be, but there is some reason to hope. There are numerous efforts being developed that would deliver pathogen sequencing in a geographic distribution that does not only align with wealth. There are many contributors to sequencing in low and middle-income countries. Africa CDC is playing an instrumental role, as has team science. We've talked about team science already, but this has been unprecedented in my own experience. For example, the recent work published in Nature that describes SARS-CoV-2 variants in time and space across Africa involved well over 100 research groups and numerous organisations. So I think this represents a climate of positive action and working together. And I would congratulate everybody in Africa and elsewhere who are coming together to make that happen. This can make a difference now, but in the longer term, there needs to be an integrated, trusted international system where member states can work with WHO and regional health organisations to generate and analyse genomic data for pathogens causing current and future pandemics. Issues such as standard methods and protocols need to be fully embedded in these efforts together with data sharing agreements. There are actually many organisations working in this space at the moment to think about how to do this or to support capabilities, including the Rockefeller Institute, the Milken Institute and the Gates Foundation, and more besides. And in addition, plans were announced by the UK Prime Minister in May 2020 of a global pandemic radar. However, it's very pertinent to mention now the funding and the development of the WHO Hub for Pandemic and Epidemic Intelligence based here in Berlin, which will strengthen intelligence by striving for better data, better analytics, and better decisions. Now, sequence data will make an important contribution to their capabilities. So we really live in a time where technological innovations can make a tangible difference to the way we detect and control pandemics. But reviewing how accessible these technologies are reveals a real gulf of inequalities. So as a global community, there's a great deal to do to change that situation, and gatherings such as these are going to be instrumental as discussion forums to highlight the great challenges and actually find ways to come together to overcome them. So thank you for listening.
Thank you very much, Sharon. And
Stay here for the question.
yeah, please, I'm just going to ask one follow-up question, if I may. You mentioned the inequities in access and also the recent African effort that was published, but I wonder if you wouldn't mind commenting on the building of capabilities. Which are the regions that actually would you say would need to be a focus, or a central area where more capability is still needed and isn't in the center?
Well, if I had one region, and it is a very large region, I would say I would go back to Charles's point and say we should be enhancing our capabilities in Africa. This has low vaccine coverage, which means that there's a risk of new variants of concern emerging and ongoing infection. And furthermore, it's a place where there's actually very patchy availability of sequence. So that would be my first choice. You'd then say, well, whereabouts in Africa? Because of course, Africa is a very big place. But I think that that's where I would zoom into. And in fact, many of the efforts people are talking about are focusing on Africa at the moment. But, variants are going to arise anywhere and everywhere. And it's really difficult to predict where they're going to happen, other than the fact that they're likely to be in places with uncontrolled transmission.
And then just following on from that, what you described and so elegantly led in terms of the building of an infrastructure that can bring together distributed efforts, how much of that and the lessons that you've and others have learned is transferable to different settings and could hence accelerate this?
Yeah. Well, I think it's right that we need to bring together an international effort, but I think that's built country by country, actually. So if every country focuses on its own capacity, bringing together its own data for its own population, then I think that could be aggregated. The lessons that I've learned, I think it's that I've learned so many lessons in the last 18 months. We'd be here all day, actually. But I think that thinking outside of the usual framework of how we work has been really important during the pandemic. We've learned so much. I think working with academics, working between public health agencies and health providers and research is so important. And that's because really research drives the improvements that you need to make the technology better, faster, and so we've had teams looking at how to do the sequencing more quickly, more cheaply. And so I think that working together as a team without any divisions will be an important way of actually looking at a kind of team effort to this.
Thank you very much. So I'm very honored to introduce the final speaker of the session, Dr. Soumya Swaminathan, who's WHO's chief scientist, and prior to this was WHO Deputy Director General. and Director General of the Indian Council of Medical Research, as well as Secretary of the Department of Health Research of the Government of India. Dr. Swaminathan is a pediatrician and distinguished scientist known for her translational research and work on tuberculosis in adults and children. She has been instrumental in promoting global whole genome sequencing of the SARS-CoV-2 virus, engaging and informing the public and professionals throughout the pandemic, and is a member of the new Pandemic Preparedness Partnership that Sharon just mentioned, established to advise the UK G7 presidency and G7 leaders on acceleration of vaccine development and global vaccine supply. Thank you so much for being here today and telling us about your own priorities.
Thank you very much, Claudia. And I think we've heard a lot from the previous speakers. A lot of ground has been covered, so I will keep my intervention brief, so we have some time for discussion. I think the WHO has, of course, a unique vantage point and also the unique position to be able to convene countries and representatives, researchers, academics, civil society, patient representatives from around the world to discuss and debate, on topics that are important for everyone. There's no doubt that the pandemic, I think, has really brought to the front pages the importance of not just genomic technologies, but technology and science in general. And I think the common layperson probably has a much better, if not complete appreciation, than at least is able to see what benefits technologies can bring. And clearly the tracking of the SARS-CoV-2 variants has been one of those amazing collaborative endeavors around the world with real-time sharing of data. I mean, on January 10th of 2020, we had the first genome sequences from China that were published in GISAID, and that allowed the world really to know that this was a novel coronavirus. And within a few hours of that, we had diagnostics. We had also vaccine development that started. So this is clearly setting a benchmark for how response should be in the future, and even building on this and making it even better. And Sharon mentioned the pandemic hub that's being set up in Berlin, and Dr. Oliver Morgan from the Emergencies Program of WHO is here in the audience, so he can speak a little more about the thinking that's going into that, what we call collaborative intelligence. So it's clear that genomic technologies, and previous speakers have given many, many examples of how this can help. But I think we need to look at maybe 3 different levels at which these technologies can benefit people. One is at the individual level, clearly for diagnosis of diseases, rare diseases, for finding treatments. The breast cancer gene, for example, is a good example. Sickle cell diagnosis. So there are many, many diseases for which genomic technologies today make not only early diagnosis feasible, but also can direct the appropriate treatments. And part of the reason why cancer outcomes are so different in high and low income countries is because you have targeted therapies that are now available for cancer. And even if some of these therapies are available in lower middle income countries, the diagnostics are not available. And again, I remember in February of 2020, Oliver, when we were sitting with the Emergencies Program, we knew it was a novel coronavirus. We had diagnostics that were actually developed here in Berlin, right at the Charité Hospital. The SOPs were first developed on the 13th of January. Those were made public, but only 2 labs in Africa had the capacity at that point to do these PCR tests. So clearly infrastructure and human resource workforce development are absolutely critical and have to be where we pay a lot of attention in order to make sure that these genomic technologies benefit everyone across the world. So as I listen to the discussions today, I think the issues that keep coming back are those of access. We're all talking about access to vaccines, but clearly access to new technologies which can benefit individuals' health is a very big issue, and diagnostics is something that's been neglected for many, many years. And Sharon and I worked on tuberculosis for many years. And it's amazing to think that the WHO has now a database of whole genome sequences of Mycobacterium tuberculosis collected painstakingly over many years. And I think we have something like 30,000 genomes there, which of course have phenotypic and genotypic, so you can correlate the drug resistance mutations. But when you think about 4 million genomes of SARS-CoV-2 in GISAID in a matter of 18 months, and we're talking 20, 30,000 TB genomes, and that too is not accessible. It's sitting in a database which is not really accessible to everybody, and we're not seeing the benefits flowing to people. Every patient who has suspected TB should have a molecular diagnostic test at the point of care, which tells them what drugs they're resistant to so that they can get on the appropriate treatment. And the World TB report for the first time, has in a reversal of gains made over many decades. There's an increase in deaths due to TB in 2020 and a reduction in the number of people being diagnosed. So on the one hand, of course, we've had a lot of the resources going to COVID, but COVID has also enabled the building of new infrastructure and taking technologies like genome sequencing to places which did not exist before. We need to ensure that those are now leveraged to help patients with other conditions. So I think we really have to think as a community on how do we make these technologies more affordable, more accessible, simplify them so that they can be used, at the point of care. So companies and innovators who are thinking about developing these technologies really need to think about a robust equipment that can work in places where there may not be electricity. So does it have a battery backup, using digital means to transfer that information in real time to a database, and also making sure the patient benefits ultimately, so that the results also need to feed back into the patient's care. So if that is done, then I think people will begin also to see the value of sharing data. And I've been listening to some conversations around data sharing now, which often becomes very polarized. But really, I think everyone accepts the benefits of data sharing for advancing our knowledge and our understanding of many different health conditions. And also to be able to use developments like artificial intelligence. You cannot use AI algorithms unless you have large representative data sets, which can be used both to train and then validate these algorithms. So again, whether it's rapid drug resistance detection of MTB using an AI algorithm, whether it's automated reading of chest X-rays, or whether it is the directed therapies for cancer, all of these algorithms need to be trained on representative data sets, and that can only happen when you have data sharing in place. So I liked also Bartha's comments about, she really brought the issues out. And I think if you talk to individuals, they would be willing to share their data, provided their data is used in ways in which public health is advanced and that they and their other people like them benefit. And secondly, they would like to be assured that the data would not be used for, say, commercial interest without, without their consent or benefits flowing back to them. And I think this, the balance between data sharing and access to benefits or benefit sharing has to be addressed because of the historical issues that we've had in this space of both patients, data being used for purposes for which they knew nothing and for which they received no benefits. And even most recently, I've heard researchers from Africa, point out that while they've been very good about uploading the genome sequence data of SARS-CoV-2 onto public databases, so the whole world knows what viral evolution is happening, and vaccine makers can start tweaking their vaccines to emerging variants, the benefits of those vaccines haven't flowed back to those countries. So there's a genuine question that's being asked here as to, this has to be a two-way. Yes, data sharing is important. It will allow us to make advances in medicine, which will help people. But will those advances eventually come back, or will people in some countries have to wait for decades before they see those benefits? So at the WHO, this is always a debate and a balance that one has to strike, and it's a very fair question, I think, to ask and that we need to answer as a global community. So we will obviously work around the governance issues, because I think they're so important that we move into an era of more open data sharing, but with these provisions that more collaborations and data sharing will lead to advances that will then be equitably shared with everybody who's contributing to those advances. So we have ethics groups that are looking at this. We have, again, expert groups that are looking at, and I'm sure the Berlin Hub is also going to advance thinking in this area, of course, related to pathogens, particularly of those of epidemic and pandemic potential, but I think the same principles will apply. And similarly, there's the issue also of researchers also feeling that, the value that they bring to the table. And again, it's rooted in historical issues of being left behind or feeling that you get left behind, because researchers from the global south have been slower to be able to publish their findings, and so therefore to get credit for the work that is done. So the WHO set up a science council. So Dr. Tedros set up a science council towards the end of last year that's chaired by Professor Harold Varmus, and that also has Edith Hurd, who's the head of the, we have several geneticists on that, Mary Claire King and others. So the first topic that they decided to focus on is genomics. And we will start a series of workshops starting in early November, going through till late December, 3 or 4 workshops that will basically look at the potential for genomic technologies to advance health, particularly in low and middle income countries. What are the challenges, opportunities, potential barriers, and what are the social, ethical, regulatory issues? Because there are regulatory issues in many of these countries which we will need to address, and some of the regulations, are not haven't been updated recently to really account for these advances, where data can just flow between people so quickly. But also there are new approaches like the federated approach to data sharing, the use of blockchain, for example, and the swarm technology that I recently heard about, but it sounds really a very attractive option, where data stays at the place of origin, but you can use blockchain to capture the learnings and help to improve these AI algorithms. So hopefully the series of workshops will give us a better understanding, and the Science Council will then prepare a short report to the Director General on how to proceed further, and perhaps what areas WHO should be focusing on. So I think I will stop with those comments, Claudia, but happy to take on anything else that comes up.
Thank you very much. It's great to hear of this set of workshops, and I'm sure they will address the question that I'm about to ask, and it's possibly not an easy question, but in terms of what you would anticipate, what would have the highest impact in terms of highest impact genomic initiative to improve health in the global south in the next few years, like not 10, 20, but 3 to 5 years. I know you mentioned TB and access, equitable access, but what would you put in the spotlight?
That's a tough one, but because there could be so many examples, but I think certainly amongst infectious disease killers, TB is number one, and it kills 1.5 million people year in, year out, year after year, so yeah, and we tend to not pay any attention to that because we've had TB around for millennia, but it is a big killer. So TB, malaria to some extent, because we've started seeing now artesunate resistance, not just in Southeast Asia, but also in East Africa. So we really need to track what's happening with the resistance to malaria. And this impacts millions of people every year. So I can see a few of these pathogens, which are, not the re-emerging infections, but the established ones. And then, of course, the emerging infections. Influenza, we've had, for decades, global data sharing, and it informs the vaccine development every year. So one could think about that. In terms of host genomics, I think cancer is probably the the one thing that comes to mind. But you know, sickle cell, for example, kills people without their even being diagnosed as having sickle cell in Africa as well as in Asia. So having point of care genomic technologies which can detect sickle at birth, I'm sure could also save lots of lives and lots of morbidity in young children. You know, it's a very painful disease to have if you don't have access to the right kind of treatment. So, and then of course there are the rare diseases. So I could think of several, and then at the population level, of course, it helps also to know, you know, what genes are predisposing people to what kinds of diseases and so on.
To the sickle cell example, that something I have read, that the vast majority of sequencing that happens in children, vast majority are born in Africa with the condition, yes. But the vast majority of genomic research is still conducted in babies of European ancestry. Is that still the case, or are there initiatives underway to address this? Maybe.
Well, I'm sure Charles can add to this, because there's a lot going on in the US. But I think one of the other initiatives that we have is really trying to look ahead to see what's coming down the pipeline in terms of you know, therapies like gene editing. And so we have also put together a group that looked at the ethics of gene editing, both somatic and germline gene editing, and put forward a governance framework for how countries might want to regulate both research and clinical applications. But yes, you're right about the sickle cell. I mean, most of the research is not coming from the high incidence areas of the world. And there are lots of misconceptions around how sickle cell behaves. I know from India, for example, people thought sickle cell in India was a mild disease that had a different phenotype than the sickle that had been described from the US, for example. But that's because most children were dying before they could ever be diagnosed. And so the ones who survived, it appeared as if they had a mild phenotype. So it was only when you did prospective cohort studies diagnosing children early in life and following them up that you realized what the real natural history of that disease was.
And that is also something I think the pandemic has highlighted, how kind of robust and rigorous epidemiology is kind of needed alongside some of the other newer technologies where we implement them. So let me just thank all the panel members again, because we're now going to have a joint discussion opportunity to ask questions. So thank you very much for your contributions. And please, first question. Please introduce yourself.
Yes. Thank you. My name is Casimiro Vizzini. I'm the Secretary General of the European University Alliance for Global Health. But I'm here because I couldn't prevent the urge of mentioning Professor Richard Cotton. Professor Richard Cotton back in 2006, created the Human Variome Project well before the GA4GH. And he had a broad vision to contact UNESCO, where I was working at the time, to involve developing countries in this initiative. This discussion I've heard now, we did almost 10 years ago. So we are still at the same point. The Global South is not involved. We, in 2015, invited Professor Raj Ramesar from South Africa to talk about during the biennial meeting of the Human Variome Project in Paris, in UNESCO, to talk about the initiative. And we're still not having that. Why? Because it's money driven, and the Global Alliance is fundamentally American driven, and engulfed completely the Human Variome Project that disappeared for lack of funding, unfortunately. But even the WHO at that time was not really interested in collaborating with UNESCO when I was the responsible of the Human Variome Project, and we were fighting and struggling to have this global approach to non-communicable disease and sharing data. I actually invite you to read an article I wrote in the AAAS publication, Science Diplomacy, about the Human Variome Project in March 2015, about the importance of the Global South and Africa in this effort. So still in 2021, we are at the same point.
Thank you for your comments. Would anybody like to?
So I know Dick, and it's very sad that he's not with us anymore. And he was a visionary and saw many things very well. GA4GH is global. In particular, it's got a driver project of H3 Africa, led by Nikki Mulder down in South Africa, but also Japanese involvement and other people. But it's not a done system for sure. And I think it's really important to have a very, very open, collaborative approach around the world for these things, which is what we try and embody. So I am sorry that Dick's not with us anymore, because he had a vision, a foresight there. I think there were some aspects about the Human Variome Project that just simply wouldn't scale. And I think that's one of the technological aspects of that era that Dick put together that wouldn't scale in that time. I'm pleased that we have colleagues such as H3 Africa, GEM Japan, and other people from around the world in the Global Alliance, but it's not a complete system at all. So I welcome more people to join in the future.
Thank you. Please.
Hello, my name is Elizabeth Mcintyre. I'm a diagnostic cancer and hematological cancer practitioner, and I work at the Université de Paris in Paris, France. My question is two-stage, and I've understood that we're in, my first question is, are we going to continue to move genomic data to hubs, or are we going to concentrate more on controlled access to primary data where it is generated within a clinical context? And if we're going to evolve towards controlled appropriate access to primary reliable genomic data, is it going to be possible to work with all the hospital directors who are going to see this data as being part of their responsibility? And is that going to get easier with time or more difficult?
Is this maybe you know, Bartha?
I don't know if Bartha wants to do that on me. So I think, so let me start by just reminding you, I think there's a very big difference here between human data, human-related data, including the metadata on infectious disease, and the data that's associated with the organisms, viruses, bacteria, and other organisms that don't have legislation and ethics. For the human data, I do think the direction of travel is responsible access, federated access. I think there will be aspects of the metadata for infectious disease, which is what Swami you mentioned, and Sharon has pointed out, where we will also need federated access, but they are quite different things. And I think it's up to each system to organize themselves. As Sharon mentioned, it's sort of up to each country to organize the way they structure that. I think in the French system, I would introduce you. I'm sure you know Jean-Francois Deleuze and all of that, and Plan France Genomic. Each system has to organize itself to allow responsible access, and so then we end up with a collaboration of 191 things, and not however many million hospitals in the future.
Thank you.
Yeah, I'd like to respond to that. The problem you're describing in France is endemic everywhere. The preference is for controlled access because of the perceived limitations of open public access with respect to data sharing that might involve identifiable data. So if, never mind that I don't agree with it, if that is the approach, what we're finding now as genomics moves to the clinic, that hospitals are sort of like castles with moats around them, but no drawbridge. And they sort of feel like they own their data. And sometimes data can't even be shared across the street. And so if controlled access is going to be the way of the future as we move slowly, if federated, having data stay where it is and people go to it, we need to do two things. We need to have common portals, so you don't have to go geographically hopping from location of data to location of data to location of data, common portals. And perhaps we should do what we're starting to do for research ethics review, is to get common DACOs, Common Data Access Committee, so that portal would not only facilitate getting access to distributed data, it would also facilitate the governance of the data with one review for access for a particular project. So we need to harmonize, simplify, and perhaps distribute federated data, but centralize some of the governance processes to make it more efficient and useful for the clinic.
Thank you.
Thanks very much. And I think as we're entering the last 5 minutes of this session, first of all, I hope you agree that we had the opportunity to hear a fantastic set of thoughts and innovative ideas. And I would like to conclude with a question to all the panelists, which really is, having heard this debate and all these thoughts, what would you really want us to talk about next year? And what do you think we should put on the agenda? So maybe Bartha, I could come back to you to start off with, then Charles, and then come back to the room.
Yeah, I only hope for two things. And one is that we start working on a pandemic treaty and get discussions going. And in the meantime, like tomorrow, activate the international health regulations of the WHO to be more powerful in the sense of being able to contribute to global public health.
Great. Thank you. Charles?
Yes, I would like to see that maybe in your next conference to highlight global examples where data sharing has definitely had an impact. I think with that kind of example, we will begin to get the attention of governments and scientists around the world. So we need to really demonstrate that the fact that we're able to have data from different parts of the world, we're able to develop and deliver on improved health using that set of data. And to, within that context, to highlight some of the obstacles that the scientists went through to be able to achieve that kind of effort, and start to see that maybe as a catalyst to other kind of data global sharing, especially if we can demonstrate improved health for areas of the world where there's this sense that, what has Genomic done for me lately? So I think we really need to, if we can do that, I will be extremely happy. Yeah.
Thank you. Ian?
I hope I get invited next year. But also I think not me. I see myself here as a representative from the basic research world, moving closer to this practicing healthcare world in both the human side and the infectious biology side. And I just think we need to move those worlds together. So I hope maybe not me, but someone like me next year.
Thank you. Sharon?
Thank you. I think I would want to see two things. One is I think the team science that continues to amaze and surprise me in a really good way will have actually gone much further, because I think bottom-up initiatives can do a great deal. People on the ground doing sequencing, getting together, reporting their data. Whilst we're in the planning stages of something top-down, I think bottom-up, sort of a grassroots initiatives are going to be very important. And so seeing the fruits of their labours is going to be something I'm really interested to watch. But I'd also be very keen to have updates on the more top-down approaches. So really hearing more about the plans for the hub in Berlin, the global pandemic radar. I'm very keen to hear how that's going to be brought to bear so that we do actually analyze data more effectively.
Thank you very much. Soumya?
I think I have to agree that we need to start talking about concrete examples of things that have worked in the areas of where genomics has really helped public health. And I think pandemic hub and the pandemic treaty, which really will bring together technology and link it to access. I think that's what is at the heart of the treaty. To see that, to see examples of where genomics has gone to clinics in places which did not have the access to those technologies before and is helping patients, whether it's communicable or non-communicable diseases. And thirdly, how researchers have been able to collaborate in a true partnership to share research data, again, to advance knowledge and understanding of disease. So in these three areas, public health, patient care, and research, one could have concrete examples. And I think if we showcase those, then it really gives an impetus to others to follow.
Thank you. And finally, let me thank the audience and the positive comments that you've submitted from remotely. And just to highlight one thing that's come up in two of them, really the technology solidarity and the kind of science collaborations that people kind of just underline, and the positive remarks about what was contributed today. Thank you, everyone, very much again. It's been fantastic. Thank you.